Memory complaints and a decline of performance of daily activities may indicate early stages of Alzheimer's disease, or AD, particularly in patients aged 65 or older. AD is a progressive disease, beginning with pathological changes that occur decades before clinical symptoms, leading eventually to the development of mild cognitive impairment, functional symptoms, and then dementia. AD accounts for as many as 80% of all dementia diagnoses. Today, we are able to diagnose AD in its early symptomatic stages by assessing biomarkers and by using tests to evaluate cognitive performance, functional impairment, and behavioral symptoms. To better understand the disease, we must look back in time to the pathological origins of AD within the brain. The two pathological hallmarks of AD appear at distinct locations within the brain. Plaques made up of A-beta occur outside of neurons, while neurofibrillary tangles composed of tau protein develop within neurons. Abnormal accumulation of A-beta within the brain may begin decades before symptoms appear. Amyloid beta is produced when amyloid precursor protein, or APP, is cleaved by beta secretase, base 1, and then by gamma secretase. A-beta is formed in the course of normal brain metabolism. However, it may be overproduced in certain individuals. In healthy people, A-beta is cleared from the brain through systemic circulation and local processes such as uptake by microglia. Both the overproduction of A-beta and inadequate clearance mechanisms can cause A-beta peptides to accumulate in the brain and self-aggregate into plaques. Plaque deposition is first seen in the brain's neocortex, the center of higher mental functions. It progresses to the more primitive cortex, including the hippocampus and related structures involved in learning and memory. Finally, it spreads to subcortical areas that regulate attention, emotion, and various other activities. Positron emission tomography can detect evidence of A-beta deposition and plaques. Low levels of A-beta in cerebrospinal fluid, or CSF, can also indicate pathology. Over time, the accumulation of amyloid plaques activates cells of the neuronal immune system, potentially causing a chronic inflammatory reaction that has toxic effects on neurons. A-beta may also disrupt the energy supply of neurons and induce oxidative stress, causing further cellular injury. Tau protein pathology begins to appear approximately 15 years before the onset of clinical AD symptoms. Tau is normally associated with microtubules, which maintain structural and transport systems within neurons. In people with AD, the presence of A-beta may promote the abnormal phosphorylation of tau, followed by its detachment of microtubules. Tau subsequently associates into insoluble structures called paired helical filaments, which further aggregate to form neurofibrillary tangles. Higher abnormal levels of tau can be detected in the cerebrospinal fluid. While neurofibrillary tangles may appear in the brains of people with other neurodegenerative diseases, there is growing evidence that the toxic effects of tau and A-beta may amplify each other in people with AD. Eventually, the underlying pathology of AD causes synaptic dysfunction and neuronal loss. This results in cortical thinning or atrophy, leading to more advanced symptoms. As the disease progresses, it extends to different parts of the brain, with corresponding biomarker evidence of pathology. As more regions are affected, more symptoms appear. Over time, pathology spreads to regions of the brain that control basic functions, such as heart rate, leading ultimately to death. AD is often diagnosed in the mild AD dementia stage, and average survival is 4 to 8 years after the diagnosis of AD, but can be longer depending on other factors. With ongoing advances in AD research, we continue to enhance our understanding of underlying disease processes and the sequence in which they occur. We now know that AD begins with clinically silent processes, which lead to the appearance of symptoms decades later. Diagnosis of the earliest stages of AD is being aided by emerging risk factor evidence, improved screening tools that evaluate symptoms with greater accuracy, and novel techniques for the detection and measurement of AD biomarkers. It is hoped that ongoing and future research continues to help unravel the causes of AD and hopefully aids in better treatment of this complex disease.
The healthy brain has around 86 billion interconnected neurons which communicate using electrical and chemical signals. Tau protein, through binding at its repeat domain, supports microtubules needed for cell structure and to provide rapid transit pathways for vesicles, mitochondria, and cell components to the synaptic junction. Aging brain neurons lose some ability to clear waste material, which can accumulate as lipofuscin, where tau can bind and misfold. Misfolded tau can capture normal tau, causing it to misfold in the same way and form oligomers, which disrupt the synapse. Tau oligomers can transfer to other neurons and initiate the same process of tau capture, causing the disease to spread. As oligomers aggregate and grow, they form paired helical filaments and then bundles called tangles. As neurons fill up with tangles, they can no longer function properly and eventually burst, leaving behind just tangles.
Parkinson's is the second most common neurodegenerative disease after Alzheimer's. The most visible features of this disease are a slowness of movement, muscle rigidity, and tremors at rest. There are also many, often debilitating, non-motor symptoms, some of which may present before the motor problems. Parkinson's affects many areas of the nervous system and different types of neurons. However, much focus has been placed on neurons in brain regions associated with the overt motor symptoms, notably a region of the midbrain called the substantia nigra pars compacta. This region forms part of a major pathway in the brain that's critical for facilitating movement. In Parkinson's, dopaminergic neurons in the substantia nigra gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. A characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged or worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, one particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
Dementia worldwide in numbers. Currently, there are about 50 million affected people with dementia, which is going to be predicted to double in the next 20 years. And if you think about it, this is causing an enormous burden on our healthcare system, but also it's causing a society and economical problem. And what is behind dementia? I'm sure many of you know that there are many neurodegenerative diseases which are causing dementia, like Alzheimer, Parkinson, Huntington's disease, or frontotemporal dementia, which is now getting more and more attention due to Bruce Willis. But there are also many other neurodegenerative disorders which are a little bit less known. And in a population of 50 million people, about 1.5 million people will be directly affected with neurodegenerative disorders. And as the population ages, this number is going to dramatically increase in the future. But obviously, there are many other people affected by dementia, the caretakers and the family as well. So neurodegeneration is a major healthcare crisis in the developed, but also in the developing world as well. And why is that? It's because there are no disease-modifying treatments available. And many of these diseases are currently uncurable, deadly diseases. So obviously there is more and more need to understand neurodegenerative diseases and human aging as well. But why is that we couldn't solve this problem? Why is that there is no cure for these diseases? This is mainly from the huge complexity of the brain. The average adult brain contains about 10 on the 9th neurons, and each neuron has about 7,000 synaptic connections with other neurons. So if you think about when you want to find out what does not work properly in this system, this is very much analogous to finding a needle in a haystack. So then the questions that arises that how do we really test a drug before using it in clinical trials, before using it in humans? And actually, can we predict success with today's model at all?